Okay, so we have our amino acids in hand, and now it's time to construct a protein. So we'll try to dabble in, we'll try to make this heavy biology, biochemistry topic as much, have as much have as many curly arrows as possible. Okay, so this is protein biosynthesis, is what I'll call it. But you know, nobody calls it that. They call it translation or protein expression. Okay, so we have an amino acid. What are we going to do with it? Let's draw our amino acid as a zwitterion. And we're going to react this amino acid with ATP in an adenylation reaction. Well, what's an adenylation reaction? So here, I'll leave those. Yeah, you get it. It's adenosine triphosphate. Okay. Here we're going to take ATP. We're going to take ATP and do an adenylation reaction. In adenylation, what you do is you allow the nucleophile to add to the phosphate closest to the oxygen that's part of the adenosine. So that introduces an adenosine site, adenosine monophosphate site onto your molecule. So here we're going to rely on the carboxylate. We're going to add to this phosphate and push off the diphosphate leaving group. Okay, when we do that, now we have we can draw our phosphate out explicitly. There's our adenosine group. So now we have this adducted to our carboxylate side of our amino acid, and there's our side chain. Okay, so after reacting this with ATP, which was up here, goodness, that's sloppy. Okay, after reacting this with ATP, we can then engage and charge the tRNA the tRNA synthetase. So the tRNA synthetase will charge the tRNA. So the tRNA, the transfer RNA, is what interacts with the messenger RNA in the ribosome to construct the growing protein chain in our polypeptide. And so what we're going to do is charge our tRNA molecule. And so some people are really good at drawing tRNAs. That's not my specialty. I think it's kind of looks like this. I don't know. Always looks like maybe one of the states. That's terrible. But we have, at this point, we have an oxygen, and we have two OHs that lead up and into the base. So this is our 1, 2, and 3 carbon. I believe that is how the numbering would go in my mind, at least. But whether we react at the 2 or the 3, the oxygen at the 2 or 3 position of our ribose sugar determines the class of the tRNA synthetase that we're using. So there's like 2 classes. Class 2 actually favors reaction at the 3OH. So what is this reaction that I speak of? So I guess I haven't really shown a reaction. I'm just kind of guiding our eye down here. So we're going to react one of these OHs. Let's pick this OH is going to react with our amino acid that's adducted to an adenosine monophosphate, which I'm going to label as AMP. So the tRNA synthetase is going to catalyze the reaction where we do a trans-esterification and we lose the acyl phosphate. The acyl phosphate's highly activated, and then our tRNA, which looks terrible again, sorry, at one of the oxygens is going to have our amino acid. Okay, so this is our tRNA that now has an amino acyl tRNA. So this is basically an amino acyl tRNA is a tRNA that is, and that's a lowercase t in all these places here. Must be too much biology for me tonight. tRNA. This is a tRNA with an amino acid. Okay, so the amino acid is adducted to the tRNA. It's been charged, so now that it can go to the ribosome. This is going to make a polymer of amino acids. Which, of course, is a protein. All right. Okay, so we're at the ribosome now. What's happening here? Well, the ribosome has two sites for reaction. It has an A site and a P site. Let me just make sure my notes are updated. Shoot, I had a great picture of this and I lost it. Let me just pull this up, sorry. Okay, so now we're at the ribosome and we're going to have two sites. We're going to have and I made that blob too big. Okay, so we're going to have a P site. And this is the site with the growing protein chain that's going to have an O, then a carbonyl, and then some R side chain, and then an O, then a carbonyl, and some R side chain, and that gets us to the rest of our protein. Okay, so there's the P site of our ribosome. We're going to react that with the A site of our ribosome. Now the A site is going to be the acceptor site that's going to accept this newly charged tRNA. And there's codons and anticodons in molecular biology and genetics happening to kind of guide how these things land and that sort of thing. But what we have here is on the A site, we've got that thing that I just drew, which is our charged tRNA. Our charged tRNA. Now the charged tRNA within this part of the ribosome, and this I believe is called the peptidyl transferase. Maybe that's a domain. That's the part where all the action happens and we actually make the bonds to grow the protein chain. But the nitrogen is going to swoop in and attack the P site and kick off the O ester. And while it does so, this whole A site is going to come over and push out the P site tRNA. So then we have an empty tRNA that leaves at the end of this process. And that's going to give us a polymer chain that now is growing. No, I don't want to put an N there. Is now growing by one amino acid unit, where this part, this new amino acid, was up here. Okay, so what we do is we charge the tRNA to make an amino acyl tRNA. Amino because it's an amine, acyl because it's got like an acylated oxygen on the tRNA. So there's the acyl group. and then that enters the ribosome, and we take that nitrogen and add it to the growing protein chain and just kind of increase everything by 1. The protein chain keeps growing until we hit a stop codon. So stop codon tells protein biosynthesis to stop. Now what happens with stop codon? Well, we are going to take our growing protein chain and instead of adding another amino acid to it, we are going to incorporate some things that will facilitate the hydrolysis of the ester on the last tRNA that was added to release the amino acid so that we have an OH. And the result is we have an OH, an R group. I'm not going to add stereocenters. So this just keeps on going and going and going and going and going until we get to the last unit, which is going to have an R side chain and an NH2. So here is the C terminus of the protein. And here's the N terminus of the protein. Okay, probably got some of the fine details wrong. Hopefully you got a chance to see how some of this works. We're going to rely on ATP to adenylate an amino acid so that it can then charge onto the tRNA, so that it can then engage the ribosome, so that it can then accept the growing protein chain and then eventually be bumped off. And this process repeats until we hit a stop codon to hydrolyze the ester to release the protein, which can then do its subsequent awesome things. Now, what's cool about this is the tRNA synthetases that charge the tRNA. There are individual tRNA synthetases for all of the amino acids, and they're all super specialized to not make any mistakes during translation. Because if you accidentally introduce a phenylalanine when you wanted a serine or something, you're going to screw up the protein altogether. So these enzymes are highly specific. In fact, we call them super specific, which it's kind of, if you sit back and think about it, a mutation is ultimately a swap of amino acid, like let me be less precise. Let me be a little flimsy. Let me be an organic chemist talking about genetics here. Ultimately, a protein mutation is just flipping an amino acid. Now you can have these things like silent mutations, in which case it's probably better to think about these in terms of base pairs. But, it seems like accidental amino acid incorporations, because these things also have editing mechanisms, accidentally incorporating the wrong amino acid is much less of an issue as opposed to accidentally incorporating the wrong nucleotide into DNA and RNA. So mostly DNA. So it's, anyway, it's quite impressive to see this protein machinery at work, because it's not 4 letters or 5 letters, which is the genetic alphabet. It's the 20 letters of our standard amino acids. So anyway, hopefully you like this. If you didn't, at least it was quick. Okay, so I guess that's it. We'll see you later.